Good evening. My name is Melissa Barona. I'm currently a junior here at the University of Miami. I'm an environmental engineering major. And I'm originally from Colombia. I came here when I was 11 years old, so about 9 years ago. And after I graduate, I hope to go to grad school to study chemical engineering and focus on alternative energy sources.
I'm Seth Marini. I'm environmental engineering. I graduate this summer, and after I graduate, I will be job searching. So if anyone wants to hire me, I'm here.
Good evening, I'm Maggie Geraldo. I am also a junior like Melissa, and I'm also from Colombia. I came when I was little, and I will be graduating next year, hopefully in May. And instead of looking for grad school, I'm mainly focused on probably like a job in the field.
We are Team Energía. We participate in the Cuba Infrastructure Challenge, and we're here to present our project on biohydrogen as an alternative energy source for Cuba.
So why biohydrogen? The reason why we chose biohydrogen instead of different methods of alternative energy is because biohydrogen is a very clean energy source. There is no greenhouse gases from its emission. The only byproduct from its combustion is water. It can be stored, and it can be used directly as a fuel source in exchange, unlike other technologies like solar or wind that first have to be converted in order to be used. That's not the case with hydrogen, and it has a very high energy content. If you can see right there, it's 120 megajoules per kilogram, and gasoline only has 44. So it's a very good energy source. And also, you can see here are the oil reserves around the globe. If you see where Cuba is, there's actually none around Cuba. So all the oil that it's used in Cuba has to be exported from a different nation. So even though currently they don't have to pay for it because Venezuela is supplying it, you don't know what the future holds. And there's also a lot of carbon dioxide that's emitted from the transportation, and especially from its combustion.
As we all know now, Cuba's infrastructure is deteriorating and it needs significant upgrades. They need to break dependency from foreign nations, as well as come up with a reliable source of energy, and what better reliable source of energy than an available source of energy, such as sugarcane bagasse.
So bagasse is the actual waste from the sugarcane production. It accounts for up to 25% of the whole sugarcane mass. There are alternative uses for the bagasse. They use it mainly they combust it to supply energy at the refineries. The problem that we have from that is that there's carbon dioxide from its emission. They also use it in the hydrolysis industry and in the activated charcoal. One thing to note is that bagasse is very rich in hemicellulose, and that's very important for biological purposes. If you want to extract hydrogen from the bagasse, that's very good because it makes it a really good substrate for bacteria. And when you burn the bagasse at the refineries, the energy content from its combustion is 7.3 megajoules, and as I had mentioned earlier, hydrogen has 120.
As of 2010, thanks to Dr. Cerejo, sorry about the pronunciation, Cuba's total installed power capacity is currently 4,900 megawatts. Besides the inefficiencies, and the majority comes from thermoelectric plants, and 31% comes from these small generators, also known as batteries. 8% comes from natural gas, and a fraction of a percent comes from both biomass and wind farming. Of all this, 30 to 35% is lost due to its deteriorating infrastructure. Here is the sugar production trends from 1955 to 2002. And I'd just like to, I'll come back to this later. I just want to emphasize on the maximum that happened in 1990. There was about 8.4 million metric tons of sugarcane produced. And I'll get back to that later. Here is a GIS map that we developed of the sugarcane mills in the past decade. Currently only 26 remain. However, I would like to just emphasize on this by just the uniform distribution throughout the island. and how transportation of energy using sugarcane mills and whatnot from the sugarcane mills would be a lot more efficient.
So due to the need for a clean source of energy that utilizes the available resources in Cuba, we have looked at 3 different methods of producing hydrogen: biophotolysis, microbial fermentation, and bioelectrochemical systems.
The first one is biophotolysis, bio meaning life, photolysis, the splitting of molecules via bombardment of electrons. It was first discovered in 1939, but it wasn't fully understood until the late 1990s. Here's a cool picture of a photobioreactor, and it's purely for aesthetic purposes. However, it is pretty cool to look at. The first type of biophotolysis is direct biophotolysis. Certain algae and bacteria contain certain enzymes that, if reduced, have the ability of producing hydrogen. The problem with direct biophotolysis is hydrogen and oxygen get produced simultaneously, making it less inefficient at producing hydrogen. The solution to that is indirect biophotolysis. Basically, they separate this direct biophotolysis into just 2 steps. And first, they produce carbohydrates, and in the first stage, they release the oxygen. And then in the second stage, they decompose the carbohydrates, and they produce hydrogen separately, producing up to 5 times more hydrogen than in direct biophotolysis. The problem with biophotolysis is currently in research laboratories, only 0.36 millimoles an hour per liter of reactor have been achieved. But with nanotechnology and new innovations coming around, it's only a matter of time before this becomes utilized on a larger scale.
Another method to extract hydrogen from waste, in this case the bagasse, is microbial fermentation. In microbial fermentation, you have the bacteria that would actually produce the hydrogen by breaking down the organic matter. The organic matter in microbial fermentation is called substrate. And as I mentioned earlier, bagasse it's a really good substrate because it's so rich in hemicellulose and in sugars. The fermentation that we looked at is actual dark fermentation because it's convenient as it occurs continuously in a reactor and it doesn't need the availability of light. Right here, it's actually different substrates that are used by bacteria to produce hydrogen. And at the very top, we have glucose, which is a component of the bagasse. And when you use glucose as a substrate for bacteria to ferment, you get the highest amount of hydrogen in the shortest amount of time. So it's very convenient to use.
Bioelectrochemical systems can be thought of as any electrochemical system where the bacteria serves as a catalyst for the oxidation of organic matter. There are 2 types of bioelectrochemical systems. There is the microbial fuel cell, where the reactions are spontaneous, so energy is produced spontaneously under aerobic conditions, and the microbial electrolysis cell, which derives from the microbial fuel cell. In this case, the reactions are non-spontaneous, so they require an external source of energy, and hydrogen is produced rather than electricity, and it occurs under anaerobic conditions. This is an illustration of the reaction that occurs in the microbial fuel cell. First, the substrate is broken down by the bacteria into carbon dioxide, protons, and electrons. Now, as you can see here, oxygen is present at the cathode, which makes the reaction spontaneous. So electrons and protons travel to the cathode, react with oxygen, and produce water. During this process, energy is produced. Now, the microbial electrolysis cell, as I said, it derives from the microbial fuel cell, so the reactions are similar. The substrate is broken down by the bacteria into carbon dioxide, protons, and electrons. The difference from the microbial fuel cell is that no oxygen is present at the cathode, so energy has to be supplied to the cell for the reactions to occur. And when energy is supplied, the protons and the electrons travel towards the cathode, react, and produce hydrogen. There's been a lot of research that has studied the potential of using the MFC to power the MEC. Because as I already explained, the MFC produces electricity, so it has the potential to supply energy to the MEC, and then in this manner produce hydrogen without an external source of energy. These are different experimental microbial electrolysis cell designs. The first one is the traditional design that was used to just prove the concept behind the electrolysis process. It consists of two bottles connected by a tube and a cation exchange membrane that facilitates the movement of protons from the bioanode to the cathode. These are continuous flow reactors that produce hydrogen continuously, so it doesn't accumulate. And this one is the single-chambered microelectrolysis cell, which lacks a membrane, because there's been research that shows that there is energy loss involved in the membrane. So the absence of the membrane can produce more hydrogen. So we've talked about three processes of producing biohydrogen: biophotolysis, which could use solar energy to produce hydrogen, and microbial fermentation, and the microelectrolysis cell that can use organic matter to produce hydrogen. Out of these three processes, dark fermentation proves to produce the highest yield of hydrogen. So we propose this integrated design that combines dark fermentation and the microelectrolysis cell to even maximize hydrogen yield. So we assume that sugarcane production in Cuba is approximately 50 tons per hectare per year. 25% of this sugarcane production results in waste, which is the sugarcane bagasse. The sugarcane bagasse now undergoes hydrolysis, where it's going to be broken down into 30 to 35% hemicellulose, and the rest is cellulose and lignin. As Maggie went over the dark fermentation process, hemicellulose would go through dark fermentation reactor, and the rest that is cellulose and lignin would go through the MEC. And so during dark fermentation, there are byproducts that have the potential to produce even more hydrogen, but that can't be broken down by the bacteria. So the dark fermentation effluent would be transferred to the microbial electrolysis cell to even maximize the hydrogen yield. And the hydrogen produced from these two processes can be collected and transferred to a 1 kilowatt fuel cell for energy production. And just to recap, 25% of the sugarcane production is sugarcane bagasse. In our design, it is broken down during the process of hydrolysis into hemicellulose and solid matter. The hemicellulose goes through the dark fermentation, solid matter would go through the MEC, and any remaining products in the dark fermentation process would be also transferred to the MEC to extract more hydrogen. And the hydrogen is collected from both reactors, transferred to a fuel cell, to produce electricity. And looking into literature, we've collected data to estimate the volume of the reactors that would be required in our design. For hydrolysis, it would be about 500 liters. For fermentation, about 4,700 liters, and for the microbial electrolysis cell, it would be about 6,500. And about 2 acres would be required for production of 1 kilowatt, which is slightly less than what a modest home would require, which is 1.3 kilowatts.
Here's the cost analysis of our pilot design, including the MEC and the fermentation and the hydrolysis reactors. also including the maintenance and operational costs of one engineer and two technicians. So basically, it will cost about $500,000 for just 1 kilowatt. However, this is just to further, because all these studies are done on a small scale. So we want to see on a larger scale what we can do, and if production rates are better, we can hopefully implement it on an even larger scale. So the overall results, I would like to just give everybody a perspective. I know Cuba's a developing country, Florida is part of a developed nation. But by relating Florida's population and total power needs to Cuba's population, we can see that Cuba is drastically under power from the 4,900, and in relation to Florida, it's about 35,000. So back to 1990 when they had their peak sugarcane production, and most recent peak sugarcane production. If we were to use this technology and to supply energy, and if we were just to use the bagasse alone, we could potentially power give Cuba about 209 megawatts. And if we were to use all of the sugarcane, then we pretty much multiply by 4 and we can get about 834 megawatts of installed generating capacity. That would increase Cuba's power infrastructure by about 15%. And conclusion.
Yeah. So as I mentioned earlier, biohydrogen it's a clean energy source. It has potential to supply Cuba, and it will break down dependency on foreign nations, even if it's just a little bit. Combined systems such as the one we mentioned, or possibly others, are good because they could maximize the amount of hydrogen that's yielded. And our pilot scale, it's mainly suitable for like a small scale energy system. As it was mentioned earlier, the areas that need the most help in, it's not like the big cities, because that's where their main focus is at. So it probably in rural areas that are more neglected by the government. That's where this pilot scale would probably be optimal for.